Hello viewers, welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share and subscribe. Let's start. We have learned about type 1, type 2. Now we are going to do type 3 restriction enzymes. Let's learn about what is this type 3 restriction enzymes. So type 3 restriction enzymes are a lot in numbers. Okay, they are present in most sequenced bacterial genomes. You can find it a lot in the bacterial genomes. Okay, and there are a lot many of type 3 enzymes that are known. Okay, so they are heterooligomeric assemblies that comprises of polypeptides encoded by restriction and modification genes. Okay, so as we did in the previous class, we got to know what are these genes that are either it is restriction, modification, or you have one more that is specification. So here you have only two subunits, that is restriction and modification. Okay, so it requires, another thing is it requires 2 copies of their unmethylated. So as you can see here, it's written unmethylated. Okay, short asymmetric recognition sites. That what does this unmethylated here means? Means it should not be methylated. Okay, asymmetric recognition sites to be present in the target DNA in an inverted repeat orientation. So what does this inverted repeat orientation means? Okay, see, first is they recognize 2 separate non-palindromic sequences. Okay, palindromic sequences. Okay, so these palindromic sequences are, these non-palindromic sequences are inversely oriented. Okay, and that is what is said here. Okay, so till here it is understood. Let's go to the next point. Now, the DNA cleavage is by the use of magnesium and ATP hydrolysis dependent. It does not require the presence of cofactor S-agenosyl methionine. Now, understand these 2 points properly. Here it is written DNA cleavage, okay, and here it's for DNA methylation. Okay, so it requires adenosine methionine and ATP for the DNA methylation, roles on DNA methylation and restriction, and it does not require S-agenosyl methionine, which is act as a cofactor during the DNA cleavage. Okay, so as we discussed before, it has 2 subunits, that is R and M. R for restriction and M for modification. So this M, it functions as methyl transferase. Okay, and this functions in the presence of adenosine methionine. Okay, and when I say R, that is a restriction. Okay, it does not require ATP to be hydrolyzed, means no hydrolysis of ATP, and it requires magnesium. Now let's talk about the next point. So how is it cleaving the DNA? Okay, so how does this occur? Now, see, this is a DNA. Now here is your recognition site is, and this is the place where your DNA, the restriction enzymes, in this place of the DNA, your restriction enzyme has been bounded. So what happens, how does it work, is once it gets bounded, it slides. Okay, it slides along the DNA, and once it reaches its restriction complex, another second complex, okay, so it cuts there. Okay, it cuts 25 base pair downstream. It cuts 25 base pair downstream. So whenever this type 3 enzyme is cutting the DNA, it cuts about 20 to 30 base pairs after the recognition site or restriction site. Okay, so this is how the type 3 enzyme works. Now let's learn about the type 4 restriction enzyme. So type 4 restriction enzyme is methylation dependent restriction enzyme. What does it mean? That means they target modified DNA that contains glycosylated bases or methylated, or they are methylated on adenine or cytosine residues, but their specificity is very weak. And the DNA cleavage is simulated by adenosine methionine, and it does not depend on ATP. Okay, so this is the cofactor and it does not depend on the ATP. So this is all about type 4. Type 4 is just a newly known and it's very short type. Okay, so we don't have much information about type 4 as of now. Now we did about all the 3 restriction enzymes, that is type 1, type 2, type 3 and type 4. So here I'm only dealing with all the 3 types, that is type 1, 2 and 3. Let's just do a bit of comparison about all these 3. Now, let's talk. So type 1 restriction enzyme, it is a bifunctional. We also discussed it in the previous video. That's type 1 is a bifunctional. That means it has both endonuclease and methylase activity. Type 2 is unifunctional. Okay, unifunctional means it has separate endonuclease and methylase activity. Whereas again type 3 is again bifunctional. Okay, so it has both methylase and endonuclease activity. Now you have subunits. So it has 3 subunits, that is restriction, modification and specificity. But type 2 and type 3 have 2 subunits, which are restriction and modification. Okay, now it requires both ATP and magnesiums. Okay, and adenosine methionine. That's adenosine methionine I'm not writing here because sometimes few require, few doesn't. So here it only requires magnesium, but again in type 3 it requires ATP and magnesium. Now let's talk about the cleavage sites. So it is 1000 base pairs away from restriction site, whereas type 2 is at or near the restriction site, and type 3 is 25 to 30 base pairs away, just 25 to 30 base pairs away from the restriction site. Okay, one example I'll give you about it. It can be ECOB, ECOR1. So you all know what is ECO. Escherichia coli. These are the simple examples of type 1, type 2, type 3 and their comparisons. Hope you understood restriction enzymes in full detail. This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.